thanks.
Do we go ahead and get started, or do we need to look at this coral some more, or what's the story?
We've got a ship move in, watch lead.
Yeah.
You can zoom in
why don't you?
yeah,
you want to explain about that?
A bunch of times on the dot.
Yeah, I know. The ship is moving, so we're just waiting for the ship to move. We have a ship move coming on.
So that other coral is actually taken over a branch of the first coral?
It has, yes.
That's interesting.
So, Dan, do you want to explain the whole thing about the ship and the Sirius?
Yep.
Yeah, make sure you're on the Okeanos Explorer line.
Yeah, so right now we're at 2,000 meters, which isn't too bad in terms of the delay with the ship move, but when we call in a move at the surface for the ship, it does take a few minutes for the vehicles at the bottom to feel that move along the cable that they're attached to. So, the 6.8 runs from the ship down to Sirius, and Sirius is kind of just dragged around by the ship moves, and then we have a loose neutral tether between Sirius and D2, and so we're limited by that 30 meter tether attached to D2. So it takes a little bit, to get things moving, and we just had a watch change, and so the ship, we didn't have constant ship moves in while we were changing the front row. But we are underway. We'll push forward, and we'll definitely make it to the end of the waypoints here.
Thank you, Dan. Roland, tell us about the cameras.
And also,
he was telling me to do it, but I said no. You do it, Roland.
Oh, Roland,
you're the cameraman.
Do
it.
Come on, they want to hear from the expert.
Yeah. And also, if you are looking at stream 3 on the quad split on the upper right-hand corner, you'll see a small version of the ROV pilot screen. And you see the direction that the vehicles are going, but in the very bottom right of that upper right-hand quadrant, you'll see an image of the Sirius, and those arrows on it is an indication of which direction the cameras are pointed. So you can sort of see right now on Sirius, it's pointed at 36 degrees, which is a little bit out and forward. And if you look on stream 2, you can actually see the ROV as it's picked up by Sirius's camera. So you can see the ROV is pretty far stretched out. As the ROV gets closer to Sirius, the camera will be pointing straight down, and then you'll see the pilot zoom back out towards the front and then start exploring again. So you can always see how fast we're moving by which direction that arrow is pointing the camera.
So,
bubblegum coral.
Paragorgia.
Ooh,
Thank
and
you,
what's the fuzzy one again?
Oh, come on.
Chrysogorgia, not Metallogorgia.
Oh, but you know what, though? Yeah. That's related, though.
Is it? Okay.
Okay.
Right, I gotta come up. Zoom out a bit. Slipping down here.
I'm learning them too, Patty. We're learning them together.
Each other looks good. Still.
I should know these from all the times we've seen them in the Atlantic. Oh, look at that one in the background.
Yeah.
Isn't that pretty?
Oh, it is.
Yeah, watch your port skid.
It helps being a diver, though.
More
damaging. Can we do a snap zoom on that?
Go ahead and zoom in. I'll hold it as steady as I
wait,
can.
that's different.
Hold there.
Yeah. Is it? Yeah.
That's different.
That is. Okay, so. And the race is on to see who types it in first. We have a chat that we are reading that is being populated by comments from people on the shore that have been joining us all day, and they are on it.
Okay, thanks.
And Osako is still here. This is the first corallidae that we have seen on this dive.
Okay, let's zoom out. Sorry, it's hard to hold a steady shot here.
Or full wide.
All right.
And Chris Kelly tells us it's a Hemicorallium CF.
I'm going to stay a little bit above
Lauense.
Yeah, it would give
the floor here until we know we're
did I pronounce
past
that
the
right?
rock.
Close enough.
I can kind of
Close
see it
enough.
on mine,
Okay.
but not
Or at
reliably
least-
enough.
Hemicorallium.
Some returns on sonar, 20 meters ahead or so.
Ooh, sediment.
Yep.
Yep. We will definitely make it to the waypoint.
Well, don't pass up all this stuff to look at.
Should,
Well, I've been
Call up
told
and
the
ask
sediment
for
is
full
actually a lot
speed.
more interesting than it looks at first.
Sometimes you find acorn worms on it. I really like them.
I found one
Well,
of
we're
those.
going to plow ahead
I'm
to
just
the...
surveying.
Well, the
seeing
worms do
what's
that
on
too.
sonar. Got a couple of rock outcrops ahead of us.
do you want me to
Yes.
Oh,
increase
no.
Yes, you can see them in the.
This is the desert that we were
In-
looking for, Les.
So we-
Indeed it
we
is.
were looking for this desert.
We were looking for
Yeah,
it,
we
and
were.
we have found it.
We have found it. So we have little teeny tiny dunes in this desert.
Yeah, let's go ahead and speed it up.
And these are called ripple marks. And they
Bridge,
form
ROV nav.
generally
Yeah, you're getting out there. I don't know if I'm gonna-
perpendicular.
Go ahead, ROV.
These
I'm
particular
coming down.
ones are
Increase-
linear
I'll be able to
dunes
give you a
or
couple
linear
meters here in a
ripples
second,
form
but I'm not
perpendicular
done
to the,
over those rocks yet.
Thank you.
the normal
That you were
current,
just over.
Yeah.
which is what they're doing. Current is from the
We
right
come
to the
down
left,
a few meters.
and they
Okay.
are
That's fine. I mean,
parallel
I was just worried about the
to
tether.
the orientation of the remotely
Well, I'll
We're
operated
keep
20
vehicle.
meters above the rocks right now, so
Yeah, I'll keep it about over 15
yeah, 15 is that's fine.
over the ground. I mean. There we go.
Seems...
And we're about halfway done with that move, by the way.
Okay. Okay. Halfway done with the ship move, I guess.
You want to zoom in?
Sure.
Just see what we got.
What have
Yep.
we got?
It's sediment, all right.
No little creatures crawling about. Oh,
You think?
no.
It's sediment.
And
Things
So what-
swimming
what-
across
What were
the
we
surface.
hoping to discover in this desert? Or what was the, we just wanted to find it? Is
we
there
wanted
anything-
to find things that have like roots, right? What are they called? Little spicules that stick into the sediment that make it possible for them to stand up in a current like this.
Okay. Let's zoom out.
Sponges? Corals?
RV Nav Bridge,
Fish.
be advised,
Oh, we got a
I
little
just
shrimp or
located
something
a
there.
small buoy approximately 75 yards off of our port
Hold.
bow.
Okay, bridge, and you think that'll
And
pass
there
us to
are
port?
hydroids on the little
Yeah,
rock.
it should pass
Coral
us
is
to port. I
next.
just want you to be aware. I don't know if it's detached or anything
Hydroids,
else.
I have
Make sure
finally
you guys
learned.
don't see anything beneath it.
Sure, thanks.
The little
Heads
ripple
up.
marks that you see
Zoom
with dark
There's,
sides to the right
floating debris,
and
probably a buoy
lighter
that's gonna
sides to
pass
the
us
left
port.
Okay.
Do they know
are
if it's
formed
attached?
generally because
They
the
don't know.
darker fragments are
Well,
heavier.
if it's on our
and
port, we're
therefore
headed
the light ones get wafted
port,
up and
so-
over the top
I
of the
mean, we're only
ripple
moving
We're
and
moving
moving
get
only a few more
concentrated
meters.
on
Point two,
the
and then
down current
Look
side.
at the current. I mean, it's moving almost a knot. I think it'll
Whoa.
probably pass us pretty quick.
Sure, if it's not attached to the bottom or anything,
Yeah.
like super long.
Oh, it's almost certainly not attached to the bottom, but it might have something hanging from it.
Okay, we can zoom out.
Okay, that's good. Keep moving.
Oh.
Wow.
So it seems the best way to survive in a desert is to find a rock.
Find a rock, indeed. Usually it's water, but there's plenty of that here.
Wow.
You want a closer look at this? Or
I
could-
do, yeah.
Okay.
Yes.
I'm sure Chris Kelly does.
So this
This
could
is-
have some anemones.
I
and
know,
I'm
that's
looking
what
at
I'm-
these
I
white
know,
spots,
that's what
trying to
I'm looking at,
understand what these are.
Yep. Yep. I'm thinking the same
Good zoom in.
thing, Chris. We'll get a zoom in on this.
I'm going to go toward the top.
Yep, sure enough.
Zoom in.
Oh, wow.
So So I've never seen these on a Tritopleura, which is the type of sponge this is. This is a first for me.
Good.
Wow.
What's the status of our collections? This would be more for collecting for the anemones than for the sponge, I would think. But do we have our 2 biologicals, or do we want to hold off? I know we're getting short on time.
We have one.
I've been trying to find something interesting all along here to grab, but.
We have one biological sample that we've collected.
We can collect
Are these
and still
anemones
make our
known?
waypoint.
No, and it's whether there's zoanthids or anemones. I mean, I think on the Leporella, the thought by Daphne Fountain was that it was most likely an anemone in Edwardsiidae, but I really don't know. So, I mean, it would be informative, I think, if it was collected. But,
okay, how about our other biologists? We have about 30 minutes left in our dive.
How much further to the waypoint?
I'm in favor of collecting this, Chris.
Like 50 meters? 40 meters?
Yeah.
Okay. We're going to collect? Okay.
Well, if you collect it, what I would suggest you do is split the piece that you get.
Yeah, this is quite large.
Put half of it in alcohol. That would be for the sponge inspection, just to confirm it really is in the genus.
Where did you say that current one is?
And then put the other half in formalin, and that's for the anemones.
Rajo.
Okay, so can I just go for this right fork? Try to get...
Yes, you can.
Okay, and that'll be enough?
That'll be enough.
Okay.
Chris, when we were zoomed in on the anemones on this sponge, it looked like a part of the body of the anemone had been inserted or is inside the exterior of the sponge. Is that because the anemones use that as a way to attach?
Basically, have your pics of bio boxes here.
Yeah, they're growing in the sponge itself, and they're just poking their heads out to the outside. So they actually live in the sponge. And just a word for the pilot: this is going to be a brittle sponge. It shouldn't come apart. It's not that fragile, but just so he knows, it's a hard sponge, and it may very well become detached by the base when he grabs a hold of it.
Okay.
Upper right branch.
I did.
Something like that. If you could break off one of the upper right branches, that should be plenty of material, I think.
Okay. Okay.
So Chris and
so this thing is just going to flake apart when I grip it. So.
We're hoping that you're just going to be able to break off that top branch without dislodging the whole thing.
Okay.
Pilot, do you need
easy, right?
Easy peasy.
Yeah, I definitely.
Okay, so Chris and our coral people would the main thing we'd be looking for to distinguish this, to be able to tell if this is an anemone or a zoanthid, would one of the things be whether or not there was a stolon connecting them to each other?
You're on, Scott.
Could you repeat the question, Shirley?
Yes, the question was, would one of the main diagnostic characteristics to distinguish whether or not these things are anemones or zoanthids be the presence or absence of a stolon?
If you need many different, let me know.
Yeah, I believe that it would. Anemones should not remain connected to one another. However, there are also independent zoanthids, so not every zoanthid would have a stolon. So that would be one thing that would make it difficult.
Okay.
Yes, we did. Clipper. Surveyed 2. Took me a second there. So, just
You know, an
curious.
easy way to do it might be to just take both of the two lobes on the top and then put one in alcohol and one in formalin and be done with it, rather than try to very carefully just take one of the two lobes at top when they're so close together.
If I get it in the Tygon tubing there that's in the claw, I'm afraid it's just going to, if it's brittle enough, it's just going to flake apart and fall off the top of the.
So, yeah, so
I mean,
what Chris was suggesting is maybe go just beneath where they join and try and break it off at that point.
I think you should get it loosely in the fingers and twist.
But will the
Try
structure...
to break it rather than cut.
That might shatter it,
okay?
though.
Okay.
It might as well.
Well, one way to find out.
Current's only half a knot. You can chase it.
It's only half a knot? Is that what you said, Dave? The current's only a half a knot. Piece of cake.
It's going to go wafting to the left.
That's why I was asking is if we could open the drawer without knocking that rock. So
yeah, that's as far as we can go.
Yeah. So we just got to be careful. Open it slowly if we're not sure, right?
You want to close it right now? Get close and we can just back off after you have it.
I'm thinking I might back off so we don't have to try to float away with it, because if it's that brittle, it might just fall apart in the current, right? So hold on, I'm going to reset up here a little further away so we can open the box. So let's close it up real quick.
Okay.
Get far enough away where we can just open the box. So let's go for the starboard bio box, please.
Okay.
Just want to get it in there as quick as possible.
Make sure that the pin actually caught, because I extended it, but it looks like it's not. So let me go ahead and retract from right here.
Well, you should retract.
Yeah, that's the method.
So I have a question about the sample that I put in formalin for the
no. I'm going to retract
Nigerians, whatever they are.
See what's going on.
Do they go in formalin for just a few days and then get transferred into alcohol?
Right, so
Closing?
what I would suggest you do is go ahead and put them into formalin for a couple days.
I'll hold it for
and then I did a very slow overnight drip in the sink there, where you just take a jar and you have your piece of sponge in the jar.
That is interesting.
You pour out the formalin and you replace it with tap water, and you put cheesecloth with a rubber band over the mouth of it, and then do a slow drip overnight. And by the next morning, all the formalin should be rinsed out, and you can transfer to alcohol.
And Shirley, I was going to follow up with what
Meg
Daly had suggested about the anemones, that
Go
it
ahead.
would be fine.
Sorry,
anemones or zoanthus, whatever it is they
biological
are,
in the port bio box
Stand by, short.
4 or 5
buoyant
days,
enough
and
to
then
float
Pilot
away
has a
from
question
the
about
box.
sampling. Stand
Okay,
by.
thank you.
Are we worried about the specimen?
Yeah, so whenever you have a
floating away?
convenient time, a number of days afterward, that would work.
Are we worried? No, I think we're okay with.
Okay.
I think we can go on
Let me
that.
check, make sure.
Yeah.
I'll open it real quick.
Oh,
I'm going to take your camera for just a second.
Yeah.
yeah.
Okay, that one will work.
Okay.
You want to go ahead and open them all the way?
Why don't you have that
make
open
it-
for as little as possible with
Well,
the other thing in
we
it?
can crack it like that. Just,
Okay.
I mean, we had to make sure it opened, right?
Yeah, no, I understand.
Is that enough if I cut it there?
Yes.
Okay. Okay, get it open.
Got him in out.
And you're going port inner.
Port mirror.
Oh, that's all we got. Can't go any further than that.
There are your 2 pieces, very nicely collected.
Indeed.
So we collected something earlier. That's all right. Don't worry about it.
I think it
It's
might go
fine.
in the
It's
other
okay.
box.
Don't worry. It's okay. I
got
think we had poured something in port outer before. Must be kind of all the way
You got
back.
2 things in port
The-
outer.
Yeah, right. I know. That's fine. We're good.
The port
I
outer
think I can
was
tell
that
them apart.
long, it was the thing with the snail
that-
on.
Oh, right,
coiled
right, right,
around
right,
the
right,
base
right.
of the
Okay, got it.
coral.
Bring this
Okay.
more inboard.
All right.
That's
Pretty sure that's sitting on it
The
still.
worm, the worm snail.
Aplicofferin.
Thank you.
The
5
shilling
bucks.
has
Okay, we're good.
sagged, I guess.
Yeah, remember we're not bringing the...
Yep. They said they had brought it inboard, but I just brought it a little bit more inboard.
Can you bring the minis this around?
Yep.
Eeks.
Got the shrimp hiding in your wake. Or in shadow, I guess.
That is
That's weird.
interesting about the bio box pin.
Yeah, we'll have to play with it on the way up.
Thank you. Alright, take my camera back.
Okay.
Alright, how much further do we have to go?
About 40 meters. I think
40
it'll move
meters
into
to our
full distance, if you like.
Yeah, let's just follow
the
them.
final
I'll wait till you pull
waypoint.
out.
You want me to turn
And
your
we have
minis back
about
to center? Or are you
15
okay
minutes
with
left
that
on our dive.
Fine with it there.
Okay.
Well, it's mostly sediment.
Should I wait until you pull
There
out
are a bunch of rocks
Oh, you
up
can go ahead
ahead
and
of
call
us,
it in.
though.
Okay.
In the sonar screen, we can see
Hydraulics are
very
secured.
strong
0.2
returns
knots,
from
okay?
Yeah, 0.2 is
small spots. So
Great.
there must be a cluster of rock up there.
Bridge, ROV nav.
Oh, we
Go ahead,
begin
RV.
to see
We
it.
need a ship move. 40 meters, bearing 000, decimal 2
Interesting.
knots.
All right, copy that. I've
Might
got
be
40
extending.
meters bearing 000 decimal 2.
Good copy.
Can you let me know my bailout again, just in case?
Yes, stand by.
So if it never retracted, would it be able to close all the way?
Bridge bailout is going to be forward. So
Yes,
090
if it's
is
retracted.
fine.
It can jump up there.
090I.
It should close if it's retracted.
And it's definitely not extending.
RV nav bridge move has been initiated. 40 meters bearing 000 decimal 2.
Thank you, Bridge.
What's this thing down here? Actually, we have like, what, half an hour left? Is that right?
A little less. Let me check. About 15 minutes.
Oh. Alright.
We'll finish that move.
Yeah, it's not much time, Andy, but do you want to drive in some high currents?
Yeah, I'll
See what
try
it's
it,
like.
yeah.
Alright.
There you go. It's a
As
nice
long as we don't have another sample coming.
Yeah.
spongy thingy.
Alright, well, we're going to do a quick pilot swap, and then we'll...
You're going to let Andy drive in these
Lit my
heavy
Oh,
currents.
yeah.
poor Andy.
It's a challenge. It's training.
Boy, Shirley, even up here, there are more of those dead stalks, totally covered with manganese, scattered on the surface.
Yep, yep. Tara Harmaluk says, sponge, different type. I agree, Tara. Looks different to me, too. Andy, you want to zoom in this heavy current over to that sponge?
Right,
take a quick look at that.
Let's go check it out.
Ship is underway. Yep. Cool.
Correct.
All right.
I'm gonna get through these rocks.
I'll come down a little bit. Don't have a whole lot of tether, but I'll come down and should be able to get there.
Okay.
Yeah, or just get close in there. Yeah, that's probably about all you're gonna have.
Okay,
Actually.
I'm gonna sit down right here then. We'll see what we can do with the zoom in just a moment.
Ooh, maybe this is one of the survivors.
I don't think so.
Really?
I don't know.
Actually, D2's already at the dive in target.
Yeah.
Made it. Okay, go ahead and zoom in.
And is that a tunicate on the left?
It is. It is indeed.
Is that what we got?
Wow.
Oh, okay, so Chris is saying
Any
this
more
is
zoom?
the same type
Okay.
that we saw before, Treatopleura with anemones.
Okay. Oh, good.
So I'll start right down here.
There's the base.
Oh, man. I get to see
there we
the
go.
whole thing.
Can we
Oh,
just
wow, that's
kinda
kinda
slowly, tilt up there for you.
Yes, please. That's good. That'd be good, Andy. Thank you.
All right.
And what
We'll
is
just
the
walk
teeny
this
little
one.
blob to the left by the base? Did you see
Another
that?
sponge.
Another sponge.
Maybe a baby one.
So you can see the little holes that the anemones are all poking out of. So they've created these little siliceous houses, or induced the sponge to create little siliceous houses from which they're living in and just poking their heads out.
Chris, do these anemones get larger?
Yeah, sadly.
Do the openings expand, or do they just grow vertically?
It's going to suggest a nice slow.
That's what I get.
This is about as big as I've ever seen any of these things. They're always a uniform size, and they have a fairly uniform distribution on the sponge too.
Okay.
Except up at the top, which is interesting.
Right, so there's a possibility that this isn't a Treatopleura, although superficially it looks like one. It's the anemones that are getting me, and the sponge is not known to have anemones, in which case is this a weird erect Lefroyella? That genus is known to have anemones. In fact, seems to always have anemones.
And a crinoid.
And that is our maximum.
Okay, that's good.
So the main point is that sponge collection may have been a new species, is what I was saying. So both the anemone and the sponge actually might have been in a very, very good collection.
Okay. Let me know if I need to.
It was a most
One
excellent
of the things
collection.
that I took from that, Chris, is that you can possibly use the associate, the symbiont, to identify the host in some cases. And we have also relied on such things with the corals. There are some things that we know are always associated with certain kind of coral, so it helps us identify it.
Yeah, I completely agree. And in this case of this genus, Lefroyella, that's the clue right there, is seeing the anemones growing in it.
Copy that.
Tara Luke was mentioning that the anemones on the last sponge were sort of pink, and the ones that we just looked at were not. Any thoughts on that?
Yeah.
Watch the pilot. Any interest in that one that was to the left of it?
I would doubt that there are different species of anemones, but who knows? One possibility is some of the anemones are in a different reproductive state than the others.
Okay.
I believe D2 is right at the end of it.
D2 is very close to the end point.
I'll go a little further though. this guy. What are all these things sticking straight up here?
Those are the things sticking straight up are what we think are dead stalks of sponge or something covered with manganese.
Okay. How about this guy? Or you want me to just keep on pushing forward?
Did you say that Sirius is actually at our final?
Well, D2
I
is.
think we are.
We're right about
D2
there.
is right now.
We are at it. So yeah, we can look at that brittle star or sea star.
I'll
Not
practice doing that real quick.
Not sure which.
Just watch your tail end as you back up.
Okay.
Should be easy. About an hour.
Okay, let's zoom in.
Co-pilot ship move is complete.
Cool. Thanks, Rod.
And the question is, does it have tube feet?
Oh, there's your feet.
What is coming out under the disc?
Sure, we'll try it. I am drifting back and forth.
What is going on with those projections coming out of the base of the disc? This is rigid of some kind, I guess.
Cool.
I think it's replacing arms.
Tighter on that.
Does look like it.
Oh, sure, that's what it is. I wasn't getting it. That's right, Scott.
They look like arms regrowing.
It does kind of look like a spider with fangs from this angle.
Yep.
And yes, I know spiders better than that to know that it does not really look like a spider.
Uh-huh.
Interesting, though.
Okay, that's good. Thank you, pilot.
Okay, video whenever you're good. Copy that.
So those were baby arms. So this one has 12 arms.
Oops, I didn't count.
I
Did you
counted
count
11, counting the 4 little tiny guys. So I'm assuming it has 12. If it were, if it were entirely intact.
I think. Did Chris Ma tell us they were in multiples of 6?
Oh no.
We
I
don't
was
have a
just
ship move in
assuming.
right now, do we?
No.
The ship is stationary, but Sirius is probably still swinging.
Maybe it was
I
a
can follow you
Tyrannos
around.
star.
Ha ha
Looks like I
ha.
got plenty of tether for right now.
In fact, I got a turn port set up to come up anyway.
Well, Chris,
so
we
it's fine.
have Chris, we have succeeded in making it to the final waypoint of this dive.
Yay!
Oh, wait, we're going to see something very cool on this rock. What is this? What have we here?
Ooh, it looks like a giant pipe cleaner. Well, we will see.
Okay, want to zoom in? Whoops. Passed it slightly. Let's try that again. No, I can get it.
Oh, surely this is a very strange
Oh,
thing.
okay, so,
Okay.
oh, wait a minute.
I'm going to tilt up for you.
Thinking dead sponge.
Another one.
Oh, absolutely.
Yeah, I agree, Shirley. I think it's going to be a dead Euretid or something like that in the family Euretidae is my best guess, but not sure.
a nice live something.
Okay, and then what do we have right below that?
Some other.
Oh, you know what it looks?
Parasitic.
It looks like a. Yeah, no, it looks like it was a.
Looks like it was
bubble
a bubble
gum.
coral
Yeah.
that's been overgrown.
Yeah, sure does. Yeah, sure
Looks
does.
like a really chewed up bubblegum coral.
It does. It definitely does. You can see what the few remaining polyps.
The little pink things.
You guys didn't even get that joke, did you?
Well, we're just ignoring
I
you,
didn't
Chris.
get the joke, but before we get back to the joke, I just want to point out, earlier I said the bubblegum corals don't have an internal skeleton like the primnoids and the bamboos and so on, and the chrysogorgiids, where you can see that skeleton. Instead, they have this thick matrix that goes all the way through the stalk and the branches that you're seeing here. It's tissue and sclerites kind of all cemented together. So the tissue is completely interworked in there. And because of that, I believe, you can have the zoanthid overgrowing it like this completely, and yet the colony still be alive. You see those just little tiny pink branchlets sticking out. I don't know if those are new growth or the remnants of the initial growth.
That's can't always use autos.
But
So
if this were
it's good to get a
doing
feel
an
for how much
overgrowth of a bamboo coral, for
trust you
example,
gotta give it.
there would be no bamboo coral tissue underneath it. Whereas here, there is paragorgia tissue all the way underneath that yellow zoanthid.
Oh, cool.
That is
Very
true,
interesting.
yeah.
Okay.
Thank you, Scott. Cool. Yes, indeed. Interesting.
Well, you're pressing down 50 to 60%.
Exactly.
Doing same thing as joy lock. Try panning left and right, and moving closer and farther away, and you got to get a feel for the response of the vehicle from the control input.
Try a floating one.
Okay, we are nearing the end of our dive. And for our shore-based scientists, if you can stay online about 5 minutes after we come off of the bottom, we will have our planning call for tomorrow's
dive. It's a
dive.
natural thing to have happen. I mean, right now the current's pushing you, but
which is going to be to the petite spot.
The tendency is to kind of drift away from things.
Maybe
it will be.
Maybe it will be.
It'll be a petite spot.
Maybe it will be a petite spot. Okay.
One more something.
How much time left on bottom nav?
We are
3 minutes.
Okay.
Right over there?
Yep.
Okay, I'll try it without any autos. And a little bit closer.
So our goal for today for this dive was to go down and explore an area
give yourselves a little more slack.
that has been targeted for deep sea mining. And so we wanted to see if this area had manganese crusts, and boy, did it ever.
Whoa.
But we also thought that we might see some corals as well, which we did, and some sponges, which we did. I think the most surprising thing for me was to see all of those dead, the stalks of the dead sponges, and in areas these big kind of barrel-shaped structures that I really don't know what they are. We guessed that they might be dead sponges, but boy, I don't know. Would have had to have been in a much shallower sea, I think. Very interesting.
The downslope is kind of from your front port quarter down to your aft starboard quarter.
Okay.
So you're going to probably want to, you can try to touch your toe down on the sand.
We were able to make it all the way through our dive track. For a change, we usually get so excited and spend a lot of time looking at a few things, collecting some really interesting and unusual things. On this dive, we made 2 biological collections.
Toe touching in the sand.
We collected a bamboo coral, which is likely new.
Sometimes when you're on a slope, though, and you're thrusting down, you're going to get dragged down slope, so you got to get a little bit of forward thrust to
which is a predator, and it was working its way up this bamboo coral, munching away at it. I don't think we have any collections of Aplocophorans, so this is going to be something new for us as well. Our second collection was a sponge with an Idarian, either an anemone or a zoanthid. We're looking at an anemone right now. And it could very well be a new species of sponge as well. So we're interested in being able to describe this relationship and whether or not these cnidarians that are on that sponge are anemones or zoanthids. And we're looking at an anemone
Daisy chain
right
looks pretty
now.
good today.
Yep. It's doing it in the rain, I think, really helps.
Yeah, Sean, you did a good
It's
job.
time to
Nice.
go, guys.
All right.
Okay, come on out.
Okay, we are leaving the bottom now,
All right. So
and Patty Fryer and I
Nice
would
job,
like to
workshop.
thank you at
so if you
home
look at your turns,
for joining
how do
us
we need to set
today
up
on
to
this
get into tow
dive
configuration?
to Friar
a
Gio
negative
in the
turn,
Marianas
ROV, and a
Trench
positive
National
turn.
Monument. And we
So
hope that
we
you
have
will
to
join
head
us tomorrow
port
for
on
another
Sirius
dive.
eventually,
We will be on a
and
place,
starboard on
we're planning on being
Well,
on a place called Petit
do we need to
Spot.
turn starboard on D2?
I
And
guess
that'll
technically we
be,
don't if we just
I think
turn
we're going to
port
be doing
on
both
Sirius.
a
Right.
benthic
So
dive and a
I'm gonna
midwater
turn
dive
port, line up at the ship,
tomorrow as
and
well.
all you need to do is
Tomorrow
turn
is a very
like
deep
20,
dive.
30 degrees
Very deep
port.
dive. So we'll,
Okay.
we're starting
Line up the ship,
we'll be
and
starting
we'll be
later
set
than
up.
usual. I mean, we'll get in
That's
the
it.
water at the same time, but we'll be on the bottom.
Yep.
Probably
So we
won't be
wanna
on
come
the
up off
bottom until about 3 hours after launch, 2 or 3 hours after launch. Okay, and for our shore base,
Got
so I want to
liftoff.
thank all of you at home
Let's go.
again for joining us. And we'll see you tomorrow. And for those of you,
So I'm gonna get your
our shore-based science
swing
team,
arms
thank you
enabled
very
here,
much
or,
also for
enable them
participating
stowed.
in this dive,
Do
for
you
providing
hit
commentary,
auto-heading?
descriptions, explanations of
Do I
what
use
it is that
auto-heading?
we were seeing
Once I'm
today.
lined up?
And please join
no,
us
because
in
once you're in
about
tow, I mean, you're gonna
5
once you
minutes
hold
to
there while you
plan
get
our
stuff
dive
stowed.
for tomorrow. Thanks very much.
And hold on 1 second.
Oh, I'm not meaning to go anywhere.
Yeah. Okay. Whoop. It's very curious. All right, so I'm going to come up a bit.
Okay.
Why don't you back up?
Backing up.
You're at a good depth. Yeah, just go ahead and back up. Yeah, now you're tugging me.
We keep getting COG for heading on Sirius.
What's that?
We keep getting this COG for heading option on
It's
Sirius.
high-packing.
On high pack.
Yeah.
Yeah, keep backing up, and I'm going to turn. And yeah, that's fine. Hold there.
Holding here.
Now you need to watch your heading.
Yep,
you need
I'm
to keep
turning back to,
lined up with.
starboard here.
Yep. All right. So now that I'm pretty much there, you can kind of start pushing out lightly and coming up, and we're going to look for ourselves in the tether cams.
Okay, coming up, pushing out. And
Don't,
you say
don't push
lightly?
out too hard. Yeah, just a little
30?
more up than out, because we're already pretty stretched out.
More
So
up.
you can push out lightly, but yeah. I can see you in my sonar, looking for the Sirius lights, the D2
Okay.
aft rail.
See him starting to come in there.
Yep.